coming to the flower. So first, the flower is defined as a highly modified and condensed reproductive shoot of plant. See here. First, I would like to explain about calyx. This is the outermost wall of a complete flowers. Its individual member is called sepal. Okay. See here in this diagram, please. This is the outermost wall of a complete flowers. Its individual member is called sepal. The sepals are generally green in color and are more or less foliaceous in texture. Calyx protects the other floral parts in the bud condition. Okay, so this is the calyx. After that, corolla. So the flowers owe their charm mostly to the bright and gorgeous color and attractive forms of corolla. So pink colors are there. That is the corolla. The individual member of corolla is called petal. The main function of petals is to attract the insects for pollination. Okay, so the main function of corolla that is attracts the insects for the pollination. This is corolla. Individual member of corolla is called petal. Okay, petal or corolla, and calyx or sepal. Okay, so coming to the androecium, this is a collective name of the male reproductive structures. The stamens may be arranged in one or more whorls. Each stamen consists of an elongated stalk, the filament bearing at its end a fertile spore producing structure called the anther. Okay, so this is the collective name for the male reproductive structures. So first, scientists given that these are the male reproductive structures. So that is elongated. The stamen consists of an elongated stalk. The filament bearing as its end a fertile spore producing structure. It's called anther. So in the stamen structure, that is elongated stalk and the anther present. Okay, see here this one. Okay, in majority of angiosperms, the anthers are bilobed or dithecous. Each anther lobe consists of two pollen sacs. Okay, so that anthers are bilobed or dithecous. So that means bilobed or dithecous. Each lobe consists of two pollen sacs or microsporangia. Microsporangia. So pollen grains or microspores are produced inside the microsporangium. See here, this is the anther, this is the filament. In the stamen structure, that's the filament and anther. Okay, in this diagram, it clearly shows that stamen structure, and hence stamens are morphologically equivalent to microsporophylls. Okay, that is about the androecium. And next, coming to the gynoecium. Okay, gynoecium. The female reproductive structure in a flower is gynoecium. It consists of one or more free or fused carpels. Each carpel is an ovule bearing modified leaf and is morphologically equivalent to megasporophyll. Androecium equivalent to microsporophyll. Gynoecium is morphologically equivalent to megasporophyll. Okay, note down. And after that, a typical carpel consists of a basal swollen ovary with a terminal style. It's surrounded with the stigma. In certain cases, the floral leaves arise in a spiral manner over the large receptacle. Such flowers are described as spiral or cyclic. Okay, so see here, this is a gynoecium, and the style and stigma is there. Okay, so in the ovary, ovule, the middle part of the yellow color is called as gynoecium. Okay, all of you clear? There is a flower structure. Okay, so in the dicotyledons, the numerical plant is pentamerous or tetramerous or sometimes dimerous also. Papaver family, we observed that dimerous. So in the dicotyledons, we are observed that pentamerous, tetramerous or sometimes dimerous also. In the monocotyledons, the condition is characteristically trimerous. So a flower which bears all the four whorls is called complete. So calyx, corolla, androecium, gynoecium all are arranged with the four whorls is called complete flower. Okay. So generally in the dicotyledons, that plant is pentamerous. All are in 5. Okay, pentamerous. Tetramerous means 4, and sometimes dimerous also. Dimerous means only 2 are present. 2 calyx, 2 corolla. That is Papaver family. Example for the dimerous. That is rare. Only Papaver family we are observed that. When any one or more of the above words is lacking, it is called incomplete. So anyone is lacking, anyone is absent, that is called an incomplete flower. If a flower has both the stamens and carpels, it is designated as bisexual. Okay. So the flower has both the stamens and carpels, it is designated as bisexual or hermaphrodite. So bisexual. And if only on whorls is present, the flower is called unisexual. Okay, only on whorls is present, so that the flower is called unisexual. A flower with stamens only is called staminate, or male with the carpels only it is termed as pistillate. Okay, only the flower having the stamens, that is called a staminate flower. Otherwise, that carpels only, it has the carpels only, it is termed pistillate. Okay. Female flower. A flower is said to be neutral or sterile if it possesses no functional stamens and carpels. So the outer 2 whorls, the sepals and petals, constitute the sterile appendages or the essential organs, as they are not directly concerned with the process of reproduction. So these androecium and gynoecium, these are the sexual parts. Okay. This is the parts of the typical flower. So, and we are coming to the common terms in the embryology. So, which terms we are very used in the embryology? The terms, different terms are there. First one is a microsporangium. Okay, please note down some important points. Microsporangium. So what is a microsporangium? The sporangium inside which microspores are pollen grains. Okay. So the sporangium inside which microspores are we are called pollen grains. These are produced is known as microsporangium. Okay. Typically an anther is 4 sporangiate. That is microsporangium. So which are having the pollen grains, that is called microsporangium. It's very simply. Okay. Next, coming to the endothecium. Endothecium, the layer of cell present inner to epidermis in anther wall. So in the anther structure, that is the layer of cell present inner to epidermis. So outer layer is called epidermis. Inner to epidermis, that layer is called endothecium. That is epidermis in anther wall. Inner to epidermis in anther wall. The cells of this layer develop fibrous bands and help in the dehiscence of anthers. So this helps in the dehiscence of anthers. That is the endothecium. Okay. All are you understood what is the microsporangium and endothecium? Okay. So next, coming to the tapetum. It's very, very important in the anther structure. We have observed this tapetum. This is the innermost layer of the anther wall. Below the epidermis, that the anther wall, that is called endothecium. Okay. That cells, that layer develops the fibrous bands and help in the dehiscence of anther. Okay. So tapetum, this is the innermost layer of the anther wall and completely surrounds the sporogenous tissue. So tapetum is of considerable physiological importance because all of the food material passes through it. This is the tapetum is the food material actually. Okay. It may be amoeboid or secretory type. 2 types of tapetum is there. Okay. So what is the amoeboid tapetum? The walls of the tapetal cells break down very early in the development. The protoplasm of these cells move into the anther cavity and fuse to form a tapetal periplasmodium. Example, alisma and butamus and typha. Okay, amoeboid tapetum. So some cells, the walls of the tapetal cells are break down and very early in the development. So that's why the protoplasts of these cells move into the anther cavity. Okay, after that, fuse to form a tapetal periplasmodium. This is a actually this is a food supply structure. So food material passes through it. It's very important that tapetum is two types actually, the amoeboid tapetum and secretory tapetum. So amoeboid tapetum, the walls of the tapetal cells break down very early in the development. So then after that, the protoplast of these cells move into the anther cavity. So then fuse to form a tapetal periplasmodium. Example for that, amoeboid tapetum, alisma, butamus and typha. Okay, alisma, butamus and typha. Next, coming to the secretory tapetum. Secretory tapetum, in this type of tapetum, the cells remain in their original position throughout the microspore development. So in that amoeboid tapetum, the tapetal cells are break down. But in the secretory tapetum, the cells remain in their original position throughout the microspore development. The cells secrete food materials from their inner phases until they break down at the time of the pollen maturity. Okay, this type of tapetum is more common all among angiosperms. So secretory tapetum is common. Okay, all angiosperms we are observed that only secretory tapetum. But some plants, that is alisma, butamus and typha, these plants having the amoeboid tapetum. Remember this, it's very important. And after that we are coming to the microsporogenesis. What is the microsporogenesis? The formation of microspores from microspore mother cell through reduction division is called microsporogenesis. See here, what happens? That formation of microspores. That microspore mother cell, it is a diploid cell through the reduction division is called microsporogenesis. Microspores are formed from the microspore mother cell through the reduction division. It's called microsporogenesis. So usually each pollen mother cell by the meiotic division gives rise to a group of 4 haploid microspores. Already you know what happened, that mother cell going reduction division when that mother cell going to divide and 4 haploid microspores are developed. These aggregates to 4 microspores are referred to as microspore tetrads. We are called 4 or grouped haploid microspores are called microspore tetrads. Okay, so then pollen tetrad, generally that 4 microspores within a tetrad are arranged, that is in a tetrahedral or isobilateral manner, that is the pollen tetrad. Okay, after the reduction division of microspore mother cell, they formed the haploid 4 megaspores, that is pollen tetrad. We are called pollen tetrad. Next, polyspory. Generally, normally the tetrad consists of 4 microspores. Only, however, in some cases, the pollen tetrads may consist of more than 4 spores. This condition is called polyspory. Okay, when it's having the 4 haploid spores, that is called pollen tetrad. So when we are called polyspory, some cases, only some cases, the pollen tetrads may consist of more than 4 spores. Okay, more than 4 spores. So then we are called polyspory. Okay, that is the polyspory. And next, compound pollen grains. Compound pollen grains. Usually the
Madam, sorry for the interruption. Madam, screen is not moving. It is you are sharing only the image of the flower. Is it right, madam?
Yes sir, yes sir.
Okay,
Actually,
okay, madam. Yeah, okay, madam. Please proceed.
shortly after me. So, but in some plants like dry maize and drosera, the spores tend to remain together in tetrads even after their maturation and develop into compound pollen grains. Okay, that is the compound pollen grains. So next, pollen wall, that we are called sporoderma also, pollen wall. It is very rich in sporopollenin and it is resistant to strong acids and adverse environmental conditions. That is the sporoderm. The stratified wall of pollen grain is known as sporoderm. So next coming to the generative cell. So the nucleus of the microspore divide mitotically from a small generative cell and a large vegetative cell. So this nucleus of the microspore divides, it gives the two cells, that is a small generative cell and a large vegetative cell. This is a generative cell. So when that microspore, nucleus of the microspore divides and forms the two cells. So when that divides mitotically, it giving the two cells. So it mitotically means one small cell and a large cell is produced. A small generative cell and large is called a vegetative cell. So next, coming to the pollen embryo sac. Normally, the pollen, see here, this is the and mid layers, this is the structure actually. So generative cell, and after that pollen embryo sacs. Normally, pollen grains contain 2 or 3 nuclei. The microspore nucleus divides to form 8 nuclei. These nuclei get arranged into an embryo sac-like structure. So when that 8 nuclei structure is formed, these nuclei get arranged into an embryo sac-like structure. So that is a pollen embryo sac. So we will discuss in later. See here, this is the formation of pollen, how that going on. Next, coming to the ovule. Ovule is a megasporangium. The ovule is a megasporangium with 1 or 2 integuments. The stalk of the ovule is called funiculus. See here, this is a funicle. And it is attached to the base of the ovule at chalaza. See here, this is the placenta, microphyte, funicle, and chalaza is there. So the integuments leave a small opening at the apical end. It's called a micropyle. Okay, so this is a micropyle, and the nucellar tissue forms the female gametophyte, also called the embryo sac. This is the ovule structure. This is a megasporangium. It has the 1 or 2 integuments, and the stalk of the ovule is called funicle, and it is attached to the base of the ovule at chalaza, and it has the integuments leave a small opening at the apical end. See here, that is the micropyle. We are called micropyle. The nucellar tissue forms the female gametophyte. See here, this in the middle, that embryo sac is there. It has the 7 celled 8 nuclei present in that embryo sac. And after that, integuments, the protective coats of the ovule are called integuments. Okay, so which we are surrounded the ovule, that is called protective coats. Okay, so those are called integuments. Ovules may be with 1 or 2 integuments. That is the integuments. So next, then megasporogenesis. The megaspore mother cell undergoes meiosis to form 4 haploid megaspores. Usually, the megaspores form a linear tetrad. Sometimes T-shaped tetrads may be formed. Usually, the chalazal megaspore is functional and forms the embryo sac. Okay, that is the megasporogenesis. Same like that microspore sporogenesis, megasporogenesis there. And after that embryo sac, the female gametophyte of angiosperms is known as embryo sac. Okay, it is developed either from 1 or 2 or 4 megaspore nuclei. So it's very, very important, embryo sac. Okay, so it is developed either from 1, 2 or 4 megaspore nuclei. So the development of embryo sac by one only, that is called monosporic. That is by 2 cells only, that we are called bisporic, or 4 megaspore nuclei. It formed with 4 megaspore nuclei, that is called a tetrasporic embryo sac. Okay. So in the detailed, I will tell after another class. And come to the egg apparatus. The egg apparatus is present towards micropylar end of the embryo sac. It consists of one central egg cell and two lateral synergids. See here, in the embryo sac structure, that egg is there, synergids, and the secondary nucleus and antipodals. Okay, so upper three cells are called antipodals, and in the center secondary nucleus. Okay, so in that three lower cells are called, one is the egg cell and two are synergids. Okay, so antipodals, they are usually three number and are present towards the chalazal end of the embryo sac. These cells usually do not have a specific function and are short-lived. Rarely they become persistent and show pastoral activity. Okay, these are the antipodals. That upper three cells are called antipodals. These are present towards the chalazal end of the embryo sac. These cells usually do not have the specific function. Okay, that is the antipodals. And the next central cell, it is the larger cell of the embryo sac and the mother cell of the endosperm. Okay, that is the mother cell of the endosperm. This cell has two polar nuclei which remain present in the center of the cell. Okay, so that is the central cell. And after to coming a filiform apparatus, it's very, very important. Filiform apparatus. It is a prominent structure seen in the micropylar end of the synergid cell. So it is made up of mass of finger-like projections of the wall into the cytoplasm. Filiform apparatus means what, Nana? That is a finger-like projections. So that in the embryo sac, in the wall, so we can see that it is a prominent structure seen in micropylar end of the synergid cell. Okay, so it is made up of a mass cells of finger-like projections wall to the cytoplasm. Each projection has a core of tightly packed cellulose microfibrils enclosed by a non-fibrillar sheath during the fertilization. That is a filiform apparatus. Next, coming to the, it's very, very important double fertilization. Okay, what is a double fertilization? In angiosperms, the two male gametes released into the embryo sac. Okay, so two male gametes are released into the embryo sac. Take part in fertilization. One of the sperm fuses with the nucleus, that is resulting in a zygote. Okay, one is fused with the nucleus, that is the resulting in a zygote. The other male gamete fuses with secondary nucleus. One with in this egg cell. Okay, one male gamete fused with the egg cell and formed as a zygote. It resulting in a zygote, that is called a syngamy. The other male gamete fuses with the in the center cell, that is secondary nucleus. It forming primary endosperm nucleus. This is known as double fertilization, as both the sperms are involved in the fertilization. Okay, two male gametes are enter into the released into the embryo sac. So the two are take part in the fertilization. One of the sperm fuses with the egg cell or nucleus nucleus, that is syngamy. It resulting in a zygote. The other male gamete fuses with the secondary nucleus, that is we are called triple fusion. Because of that secondary nucleus, two nuclei are present. One male gamete and secondary nucleus, that is called a triple fusion. It forming the primary endosperm. That is a triploid in condition. Okay, primary endosperm nucleus. This is known as double fertilization. These both the sperms are involved in the fertilization. So that's why we are called double fertilization. Okay. All are you understood? Very, very important. Double fertilization. So next, coming to the syngamy, the union of male and female nucleus is known as syngamy. This result diploid zygote is formed. First is described by the syngamy is described by the Strasburger. Okay, triple fusion, double fertilization given by Navashin. Triple fusion, so that is the union of male nucleus with secondary nuclei is known as triple fusion. So that is triple fusion. And next one, perisperm. Generally, the developing endosperm consumes the nucellus completely. However, the certain families such as Amaranthus, Canasi, and Paberaceae, etc., the nucellar tissue persists in the seed and becomes packed with food materials. That is called perisperma. Okay. Next, coming to the polyembryony. So that the occurrence of more than one embryo in a seed is known as polyembryony. Okay. In that name, poly means many, embryony means many embryos are developed. Okay, that is called polyembryony. Polyembryony examples are citrus and mangifera. Okay, mango, citrus and mangifera. That is the polyembryony. Next, only three are there, sir. Please, only 5 minutes. Close the session, sir. Next, coming to the parthenogenesis. Okay, parthenogenesis. It is the process of embryo development in which egg cells develop into embryo without fertilization. Okay, that is parthenogenesis. It's very, very important. Parthenogenesis. Sometimes in our entrance exams, that polyembryony and parthenogenesis, apogamy, these are very important terms. Parthenogenesis means what? It is the process of embryo development in which egg cells develop into embryo without fertilization. Okay, without fertilization, the embryo will be formed. That is the parthenogenesis. Example is Taraxacum and Alchemilla. Okay. Next, apogamy, apospory, and apomixis. These three terms are also very, very important in our CPGET entrance. Apogamy, the development of the embryo from cell of embryo sac other than the egg. See here. So parthenogenesis means without fertilization, the egg cells develop into embryo. Apogamy means the development of the embryo from the cell of embryo sac other than the egg. Other than the egg, remaining cells are developed as embryo. Okay, the development of embryo from cell of embryo sac other than the egg, that is called apogamy. So there is example for this allium and iris. Okay, apogamy examples are allium and iris. Next, coming to the apospory. So in this process, a somatic cell of the nucellus, or of the archisporium directly forms an embryo sac. The diploid egg parthenogenetically develops into an embryo. Okay, hypercium. Example is the hypercium. Apospory means, so that is somatic cell of the nucellus. Somatic apospory we are called, or of the archisporium. So this archisporium directly forms an embryo sac. This the diploid egg parthenogenetically develops into an embryo. This is the apospory. Okay, in this process, that a somatic cell of the nucellus it's formed as a diploid egg. Okay, so this is a hypericium example for the apospory. And next, coming to the apomixis, the substitution for the sexual reproduction by an asexual process. See here, it's also very, very important. Apomixis means the substitution for the sexual reproduction by the an asexual process, which does not involve any nuclear fusion, is called apomixis. See here, in this, that it's substituted by the that sexual reproduction by the an asexual process. So in this process, which does not involve any nuclear fusion, that is called apomixis. These terms are very, very important in the embryology. So I think all are completed. Egg apparatus, polyembryony, parthenogenesis, apogamy. Okay, syngamy, double fertilization, triple fusion, integument, ovule, generative cells, pollen grains. Okay, tapetum, endothecium. Sir?
Yes, madam.
In the next class, I will continue
Sure.
from the anther structure, sir.
Okay. Before wind up the session, I
Okay.
will give the chance to the students, those who are
Okay.
interested to interact with you.
Okay.
Please unmute yourselves, my dear aspirants of CPGET 2024. Please interact with our resource person. Okay.
Please share your doubts, Nana. Any doubts?
Any doubt, my dear friends?
My dear students, please, any doubts?
Madam,
Please,
good morning, madam.
good morning.
Can you please repeat the point of microsporangium again?
Microsporangium.
Hmm.
Okay. So the sporangium inside which microspores are pollen grains. Pollen grains are produced. Okay, in that sporangium, that pollen grains are produced. It's known as microsporangium. I think. Are you understood?
Yes, ma'am.
Okay. Anyone, please tell me any doubts or it's clear.
And one more, ma'am.
What is your
Tapetum,
doubt?
you told that tapetum examples, na?
Tapetum.
Examples.
Amoebae tapetum and secretory tapetum. Amoebae tapetum examples are alisma. 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 And butamus. Typha.
Okay, ma'am. Thank you.
Secretory tapetum is common all among angiosperms. Okay. Anyone? Any doubts, please?
Okay, madam. Thank you. So, my dear friends, this is the introduction of our plant embryology session. So, remaining, madam clearly explained about the microsporangium and megasporangium development, embryo sacs, endosperm types, and all the details will clearly madam is going to explain next classes. So don't miss this wonderful opportunity because of madam has clearly explained about the introduction of today's class. Because of today's class is most important, especially the discovery of the all the scientists related to discovery of related to plant embryology. So madam, thank you very much. Tomorrow onwards, our embryology sessions continue. Thank you.
Okay. Okay. Welcome, sir.